So not all cells have to be spherical in shape, so they could be different cells. So you can actually see that these are some of the cells and the insides of the cell organelles. You can see all that, right? So the A is a Stratophylococcus bacteria. I'm not a microbiologist, so don't quote me if I get something wrong in here. But yeah, so there could be different kinds of cells, and they could have different cell structures, right? Not necessarily sphere, and you can, as you can see in this thing, it's like it's like a square shaped, right? So this is, but they will have some common features, and some of the common features is what we will be interested in. So I'll put these things up for some time. Right, so let's look into some of the, like, what I call rules of life. Now, life will always be made up of one or more cells. You will not have life without a cell. There is no life without a cell. You need that cell for life, at least one. And as we saw, that single-celled organisms are the most common ones. Why? Because it is simple. Simple things will always be more in number. Hydrogen is always more abundant than in any of the others. So simple things will stay simple for a long period of time before becoming complicated, because it takes a lot of effort to become complicated. Okay? It stays simple and then becomes complicated. All right, now the cell is always remember, basic unit of life. Anything that is smaller than a cell cannot be termed as life. And the third, and I think it's one of the most important ones, is all cell comes from pre-existing cells. This is very similar to the argument that we were looking into the second chapter, right? When we were looking into the maggots, the flies coming from the meat, that experiment. So what was told, that life comes from pre-existing life. It's the same thing. What I have done is interchanged life, interchange cell for life. So I'm saying all cell comes from pre-existing cells. But what will be the problem in this approach? The first cell. How did you get that first cell? It's a problem that people are trying to solve. It's a major point of research right now. How do you get that, first of all, and what conditions were required to get that? Because it cannot be serendipitous, right? I mean, it's a very complicated system. You cannot have a stroke of luck that it has to be very specific condition that, and we are trying to when did that happen? How did it happen? So it's very interesting. Okay. Now, looking into the two types of these prokaryotes versus eukaryotes. So, prokaryotes that have a prokaryotic cell, which is what? Bacteria and the archaea. And the eukaryotes, which are all complex organisms, plants, animals, even fungi. So fungi is also very complicated. I mean, so that is also a eukaryote. Now, this prokaryotic cells or prokaryotic life generation was simple. These are simple cells. These are single cells, these are simple cells. And you can imagine, I don't have to convince you that these things are complicated, way more complicated than this. The production of these must have been faster than these. And they were also very robust. Also, they are simple because you can also see that they don't have any nucleus. You can imagine that this is like a one-bedroom apartment, this thing. And this is like a factory. Maybe not as simple as that, but a one-room factory versus a multi-room factory in here. Because in this one, everything is very simple, so they don't need they don't need that complicated mechanism to go. They are simple, they were robust, and that's why their production was fast. And they remained this for a long period of time. So they remained like this for about 20 billion years. They still are around. They are not extinct. But the complicated life came in after 2 billion years of life hanging around. So they stayed simple for 2 billion years before becoming multicellular. Right? Now, after this, you get the more complicated life, and they have compartmentalization inside. Compartments, then you have the nucleus. What is the nucleus doing? Is a storehouse of what? Energy? It is that information center, right? So a separate section takes care of the information as the information center for this entire cell. So this is important, okay? You can imagine this as a protective vault for this entire information center that is getting passed along to the next cell, because cell comes from cell. So that's what is happening, okay? We see that cells can have different kinds of structures, but all these cells will have these 3 simple components. One is a membrane. I'm not going into all these organelles. And so one, they will have a membrane, okay? Now, what is the membrane going to do? It is a boundary, right? So this is the boundary. So what will it do? What comes, so this is an enclosure, encloses the chemicals, right? The second thing is, this helps in the transport of materials in and out. So anything that comes in or out, the boundary or the membrane is going to, it's like a fence of the factory. Right? So you have a factory, you have a fence, so like anything that is coming in or out, the fence or the boundary is going to take care of that. There are other functions of the cell, because this is not just a static factory. This moves, so this membrane will also help us in attaching itself to something else. So if that cell wants to attach itself to something else, this is the thing that is going to attach, the membrane. So not just the movement, but also associations with anything that is outside. It could be living or non-living. So these associations are going to be controlled by the membrane. Okay. The other important thing that we will need to know is the information center or the nucleus. What is that information? The DNA is that information, and the thing that has that information center, the nucleus. Okay? So anything that will have, so not necessarily the nucleus, but in a prokaryotic cell there will be the nucleus to hold that information. But even in the eukaryotic cell, you will have a seat that contains that DNA. Okay? Now, the third, what will be the third? Yeah, that is there, but tell me a very important thing. I mean, you can say the cytoplasm, then you can say all these cell organelles. Energy. Yes, so what is that energy seat? Yeah, right? So then you have the energy production unit, and you will have the mitochondria. Now this, most of the mitochondria that we know of relies on oxygen to generate energy. Most of the mitochondria that we know of will rely on oxygen. So all those life forms that will rely, whose mitochondria will rely on oxygen, what are we? Does our mitochondria rely on oxygen? Do you need oxygen to live? So our mitochondria need oxygen to produce the ATP. There will be certain mitochondria that do not require oxygen. That's possible. And those are the kinds of organisms that can live even without oxygen. Can you give an example of some of those organisms that can rely even without oxygen? A certain gut bacteria, for example, that stays in your gut. They don't need oxygen. So there are many organisms that require oxygen, but there are also many that do not require oxygen. We, for example, we need oxygen. Okay. Now there is also a type of cell organelle that can produce energy in plants. What is that called? Chloroplasts, right? This is the factory unit that converts sunlight into food. And this exists only in a certain type of organism. What is that called? These are the plants. Right? So we don't have these. There is also another way in which a plant is differentiated from an animal cell. What is that? Cell wall. So you will see that a cell has this membrane, which is called cell membrane, right? A plant will have an additional wall on top of this. So this is called a cell wall. And this is the inner one is called the cell membrane. This cell wall gives plant cell a kind of rigidity. Okay, animals don't have that cell wall. Okay, so this is the plant cell. The plant cell has cell wall, and they also have chloroplasts in them. Okay, very basic, but there are other differences between plant and animal cells as well. Not going into that, but let's talk a little bit more about the membrane or the boundary, because this is the place where multiple things are happening, the exchange processes, the boundary and all those kinds of things. Which biomolecule do you think that we studied in the past is going to play a role in here, in making the membrane? So we saw 4. What were they?
Carbohydrate, protein.
Yeah. Carbohydrate, protein, lipid, and the nucleic acids. Which of these do you think is going to make the membranes?
Protein and lipid.
Let's look into a class of compounds called the phospholipids, which we briefly touched upon. Now, tell me what were the lipids?
Fatty acids, chains of fatty acids.
And what is a fatty acid?
Long alkyl group, and carboxylic acid.
Do you agree? What is a lipid?
Glycerol.
What are they? The lipids? So essentially this is a carboxylic acid, but with a long carbon chain. So this will look something like this. And then you will have a COOH group. But what will be phospholipids then?
Attached with phosphate.
So there will an additional phosphate group that will be attached to it. This is how it can go. So like there will be phosphate groups that will be attached to it. And we saw the different kinds of linkages that are possible within these biomolecules, right? So this is a phospholipid. This is a lipid with a long carbon chain and the COH group and these phosphate parts. Now, simplistically, let me represent this as something like this. What is the nature of this part versus this part? So there is a particular behavior that these long carbon chains are giving versus a very particular behavior that this carboxylic group and these phosphate group, these are attributing. What is that nature? Do you see a difference in charge in these two things? The phosphate part, do they have a charge? Or the COH, do they have a charge? Or a dipole moment? They do have a charge. That COOH group can go to COO minus. That phosphate, that thing has a charge. So the moment you have these phospho things attached to it, think about charge. Think about dipole moment. So I tell you that this is charged. Now what can you tell about the nature? There is a difference. There is something happening with this verse and this. And we discussed this when we were discussing the lipids. What will this thing be attracted to? What will this thing be attracted to?
The tail is hydrophobic.
So this is hydrophobic. Hydro means water. Phobic means fear. That means this end will repel water versus this end philic. So this will attract water. Correct? So this entire thing together is called what? The same compound when it has both hydrophilic and hydrophobic parts. Amphiphilic. So you have to remember that these lipids are amphiphilic. What is the meaning of amphi? Undecisive, kind of. Think about amphibians, both water and land. Right? So amphiphilic. Amphoteric, both acidic and basic characters. The thing that shows both acidic and basic character is called neither acidic nor basic, it's called amphoteric. Similarly, amphiphilic is both hydrophilic and hydrophobic at the same time. It's the same compound, but a part of it is hydrophobic, a part of it is hydrophilic. This thing helps us a lot to sustain life. How? Let's look into this. Let's imagine that you have all these different amphiphilic molecules in water. Imagine life has not originated, but you have water in which these compounds are just hanging around. What is going to happen to them? Because of their nature, they are going to come together in a particular fashion, right? We already saw this, right? So the phospholipids and all that. This is what is going to have when you have, so this head is kind of the hydrophilic head versus this is the hydrophobic part. That's how that is represented. Right? So you can see, so this is one of those phospholipids structure, you can imagine. This is the other one, this is the other one. When you have a lot of all these things in water, because of the nature of the molecules, they will come together in this particular orientation. Because all the hydrophilic heads, they will try to come towards water, wherever there is water, and they will kind of protect its hydrophobic parts from getting, because they're hydrophobic. They've started to do what? Kind of form a structure in here, right? So imagine when life is trying to originate, or like we have a lot of water, and then you have these molecules somehow. Then all these different molecules will come together and kind of make an enclosure. At least we can imagine that thing happening, right? So that is what is happening in here. So it's all these hydrophilic things will come in the outside, and it is in the inside they are going to form an enclosure. We don't know what is happening in the inside, but this enclosure, you can imagine that enclosure being formed, right? So this thing is called a lipid bilayer membrane. Why bilayer? So you can imagine this as the first layer and this as the second layer. So this is called the lipid bilayer. Now, these lipid bilayer can have different, as I mentioned, that boundary is helping in 2 or 3 different kinds of things, right? In and out of the molecules. So it can attach itself with something else, biotic or abiotic, that membrane, a cell membrane can do that, right? So that's. The other thing is it can let food come in and waste go out, because you need that mechanism somehow. You need to eat, you need to get the food in somehow. And you have to dispel the waste products somehow. So that also has to be taken care of by the cell membrane. What else is there? That's the fence. That's the in and out, right? How do you do it? There will be certain, you can imagine them as gates in that fencing. It's not all wall. There will be certain gates that will allow the in and out. Very similar to any enclosed structure anywhere. So you have a big enclosure and then there will be gates from which cars, human beings can go. So it's the same thing. And those gates will be some kind of a protein that will allow that in and out. Okay. Now these proteins I'm just calling them proteins for simplicity, but these will be complicated channel systems that will allow that. How are they actually getting the food in and the waste out, and not the other way? Because there could be a lot of waste on the outside. How do you filter that and get only the food in? So all that things, complicated things has to happen. These proteins that will be on the structure are called the transmembrane proteins. These will serve as the line of communication between the inside of the cell and the outside. Is there anything? Okay. We'll continue this in the next class.